Okay. Yeah. So I sent around an agenda yesterday, and I think this meeting shouldn't take too long. We're going to go through the initial modeling results that Conrad ran. Based on that last meeting we had, we decided we'd focus on one section of the lead molecule and run some modeling studies on that. And so Conrad has those results. We've kind of gone through them, at least Frank and I and Mark as a group, and picked out about 23, 24 building blocks, and those have been ordered. And then I'll turn it over to Frank, and he'll just kind of go through some of this initial synthesis that is now being done in his lab. And the second part of the discussion, that's why I said I don't want to put this up on the YouTube videos right now, was just be to discuss some potential funding opportunities. There is a few calls out right now that we can consider for additional funding for this program. So we can talk about that, and then just to end off, some next steps on additional modeling. And I just actually got off a call with Charles River, and they have approval, at least at a high level, to continue on with us to do some potential chemistry for us. So if we actually get some new modeling and have some compounds for them to prepare, we can be in a good situation to take advantage of some in-kind contributions from Charles River. So I'm actually going to stop sharing my screen and give control over to Conrad, and he can go through his modeling exercises.
Okay, I don't know. Can you see my screen?
Yes.
Okay, so in this part I will talk just about what we did, the modeling, and then which is the first round of the work, to focus on analogs of this molecule based on modification on this C ring. So this this is the information we got from the chemist, what we need to search and what the building blocks that they needed. So they wanted 5 or 6 membered rings, aliphatic or aromatic, attached to either an acid functional group or an acid chloride. So I did that while taking into account all the exclusion that they requested, and search the chem space, 100 milligram sample with the price, and then got about 2,400 entries when it accounting for the C5 plus 5-membered ring with OH and acid chloride functional groups, while for the 6-membered rings I got about 4,500 entries. So the next step was to enumerate these reagents based on this scaffold, and the connection or the connecting point was on this NH. So if you see, it's just to form back the same kind of structures. So I did that, and for the 5-membered ring compounds, I had about 6,400 entries, while for the 6-membered ring, I had about 16,000 entries. I combined these entries to form a unique, a single file which I'll use for the docking. So during the enumeration stage, I realized there's a few things that were a bit of a concern. For example, reagents that came in different forms, but the same reagent. For example, we have this one that came in as a sodium salt, a neutral molecule, or with a chloride salt, and so on. So we had this same reagent appearing several times, which will increase the amount of final molecules we have in the enumerated set. And then we also had, for example, this one where we had acyl chloride, and it's the same molecule with the acid functional group. So if we enumerate this part of the molecule goes, and we'll end up with the same product at the end. So these are some of the causes for the repetition we are going to see at the end. And finally, so as I was saying, such a molecule, we have such repetitions throughout. So for every reagent, we have some type of repetition in it. And that's why you see these are the final products with repetitions. And then at the end, during visual inspection, also, I realized that there were some reagents with this type of elements. For example, in this case, silicon, which I think was not necessary. So I had to filter such of these outliers from the set. And then finally perform docking with the final set of molecules, which I thought was somehow the clean set. So for the docking process, the first step we do is usually the validation or redocking of already known ligands, most importantly, the ones that are already crystallized. And in this case, our reference was using the PDB IT6W6D because this was the closest to what I was working with. And it did dock well with good docking score. And then the docking pose had root mean square deviation very close to the experimental pose, which was validating the procedure you want to use. And then I also did redocking and cross-docking with other targets, for example, 5WCF and 6WAD, to see if molecules from these PDBs could dock in the other PDB and vice versa. And they all did dock well, and the root mean square deviations were equally less than 1 Armstrong, which was quite interesting. So because the validation was successful, I did dock the total set of molecules that I had at the end. And these are just some of the docking poses and how they aligned or superimposed to the experimental structure. So we can see that, for example, these 3 random selections superimpose quite well with the experimental structure. So this is based on the idea that similar molecules with similar structures should interact or bind in a similar way. So after docking, the next step is to evaluate the stability of these molecules. And to do that, we run MD simulation on a set on the dock poses. In this case, I ran 10 nanosecond MD simulation, writing out 20 frames. So for after a certain time frame, a snapshot is taken, and then which is used to generate these figures to see how. So the graphs are just to show how stable the molecules are. The reason for this short MD simulation is just to check if there's an early drifting of these molecules from the dock poses. And so this is with respect to the reference ligand, which did set premises for us to use cutoff values for the entire set of molecules that we ran MD simulations. And after our last meeting and then further discussions with Matthew, we did use some of these cutoff values to select 61 molecules, which we submitted to the chemist for their validation or synthesis. And these are the 61 selected molecules that we submitted. So in addition to this, we had plans to run maybe more rigorous computational work, so free energy calculations. But with the number of compounds that the chemists wanted. That could not be done because the number was extremely large for such a computation from our side for now. So we only do that with after the synthesis to optimize those set of compounds. Okay, yeah, so that's what I had.
Thanks, Conrad. Does anyone have any questions for Conrad on his modeling exercise? Okay, so maybe Conrad, you can stop
Okay.
sharing, and I'll start sharing my screen again. Yeah, so we basically looked through that list of 61 compounds and chose these 24 compounds, uh, which I have now ordered from ChemSpace. The lead time on that is around 2 weeks, 2 to 3 weeks. So we'll have those compounds in the next couple of weeks, and by our next meeting, we'll have made some of these analogs and hopefully start the process of getting these compounds tested. So perhaps I can turn it over to Frank, and maybe you can just quickly go through the chemistry that's been done to date.
Sure, yeah. So we've Jillian, who's a grad student in our lab, so she's going to be working on this a little bit part-time, and then we currently have, or recently had a new postdoc who's joined last week, and so he'll be starting on chemistry this week as well. But Jillian was able to start the synthesis. So again, starting with the aminobenzoic acid, as you see in the top scheme there, we had to make that first intermediate that the paper from SGC used as the starting material. It's just much cheaper than purchasing the reagent itself. So we had to make that first, and then Grignard addition makes the ketone. After an alkylation, we were able to make the benzodiazepine core that has the unsaturation in the ring. So we currently have about 500 milligrams on hand of that. That's currently been done on a smaller scale for now, just so they have a hand in the chemistry. After reduction, we'll have that advanced intermediate for all of our amide couplings. And then what Connor, who is our new postdoc, will do is bring much more material through in the next couple of weeks. And hopefully we'll have a couple of grams benched for that advanced intermediate so we can start making some analogs.
Okay. So I think a lot of the building blocks that I ordered are at minimum 100 mgs. So what will your target for the final compound be? I would guess 10 to 25 milligram range would be what you would aim for.
Yeah, that's usually what we do. In our other programs, we tend to aim for at least 50 megs.
Okay.
But we're flexible in that sense. If they're all coming in with 100 megs, we're more than happy to just aim for 25 and go from there.
Okay. I think, yeah, 25 certainly I think is enough that we'll be able to run, certainly any of the enzyme testing, cell-based assays, and if the compounds are of interest, we can advance those further down the pipeline for DMPK and chiral separation.
Sounds good.
Okay. I think one last comment. I know Pete, you're on the line. So from Conrad's modeling exercise, is that the sort of data that you guys would need right now, or would you be looking more for just the data that we generate from the modeling in association with the assay data?
We would use both. I mean, if we could take that modeling data as well, the positive and the negative results of the entire modeling, we could use to help train.
Okay.
Yeah, and obviously as experimental data comes in, then put that in, and then so build in all of it.
All right, perfect. So yeah, I can get Conrad or Matthew to send that data along to you, and or the modeling exercise, the readout from that, get them to send that to you.
And maybe one further question on that. One other way
Sure.
that we could try to start to explore chemical space more broadly is we could generatively suggest things, new analogs based on what we've learned so far, and then put those into the model, right? So that you slowly try to, through modeling, expand the chemical space that might be suitable. That might help us when we actually explore new chemical space to do that we call that active learning. So
Right.
you do a few rounds of that where it's basically the generative engine learning on what data is available, using the docking model to find out if these are crazy ideas or not, and then incorporating that data into the model, like 2 or 3 spins, right? Then by the time we're ready to potentially look for new scaffolds, the generative engine will be more prepared for that.
That sounds cool.
So maybe the first step is just sharing in the data that's available.
Yeah. Okay. I think, yeah, we did send you that SD file with that original data.
Thanks for that. I mean, including this docking round. Yeah.
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 Okay.
Thanks.
Yeah. All right. Yeah. So the next section, and Rachel and I just had a conversation about this yesterday.
Sorry,
is
Peter. I wanted to ask a question about
Oh,
the
sure.
compounds.
Yeah.
So yeah, it'd be helpful, Frank and folks at UHN, if you can please just like keep us in the loop, like when you think compounds will be ready, just so that we can ensure that we can have the assays run like expediently. It's just because we have to make sure we've got the sufficient radioactive precursor for the activity assay. And we don't like to keep it for too long. So yeah, so it would just be helpful if you can let us know, oh, you know, these will be coming in like 2 weeks, 4 weeks, whatever it is, then we can just make sure we're prepared. We've already purified a ton more PRMT6 protein, and we've also been backfilling our PRMT selectivity panel just to make sure we've got all of the proteins that we're going to need ready to go. But yeah, it would just be helpful for planning. That's it. Sorry.
No, that's a good point. So in terms of when you run these assays, is it best if sort of they accumulate maybe 10 compounds and send them all so that you can run it all at once?
Absolutely. Yeah, that's the best way to do it.
Okay, so minimum of 10.
Yeah, or
Okay.
something in that kind of ballpark. Yeah, I would say.
Right. So not they won't send. Well, I suppose they can send them to you, and you can just
We just won't do
once.
anything. Yeah.
Yeah.
So, but just so everyone's like aware of like what our process will be. Yeah.
Yeah. No, that's good.
Yeah, and I appreciate like it's kind of difficult to sometimes know, you know, it's a bit like asking how long is a piece of string, but just yeah, let's just keep in contact about it, and then we can just make sure we're efficient.
Yeah, I mean, there's going to probably be sometimes when you'll get 5 compounds a week, and other ones might be a little bit more challenging. So
Yeah.
I would just run your assay on what's most efficient for you.
Cool. Sounds good.
Sorry, just to quickly add on that. So we typically give, like, I can give a week's heads up, or is there any optimal kind of time that you would like? We can do whatever works best for you.
Yeah, no, that's fine. Because once we receive the compounds, we'll need to register them, solubilize them, and plate them, get everything ready. So then it'll be another week before we actually probably run the assay.
Okay.
So that's fine. Yeah, great.
Perfect.
Thank you so much.
No problem. Thanks.
Okay. So there's a few new funding opportunities that have come out, and some of them are the calls coming up pretty quickly. One of them that's kind of an obvious one that I think we will definitely put in for is this Kennedy's Disease Association. That is for up to 100,000 US a year for 2 years. And I think in order to kind of make sure that we're not overlapping on some of the grants that we've applied for already, one being the Conscience Grant, which we've been awarded for. Rachel has put in for an MDA grant as well. Not sure when we're going to hear about that, hopefully soon. But I think for the scope of this one, if we kind of maybe focus a little bit more on the chemistry side, certainly on the modeling and maybe some of the AI, along with what Pete just described, that they could do it variational. I think that could be a heavy part of the scope for this grant, and then essentially copying pasting a lot of the what we already have written down from some of those other grants. That proposal is due in a couple of weeks, so I think it's not a lot of work to put that together, but I think like to hear people's thoughts on if that scope sounds reasonable for a disease or a grant of this magnitude. Certainly $100,000 is not nothing to sneeze at, but it's not a huge amount of money either.
I mean, so I already basically have text for the Kennedy Disease Association grant that we can reuse from the previous MDA grant. Yeah, so it's not difficult to pull this together. The proposal is pretty short. It's only I think 3 pages.
Yeah.
So it's pretty easy to do. I mean, my suggestion is, particularly at this early stage, kind of it would be good to get some more funding for the sort of computational power of things and the AI modeling, all that kind of stuff, and benchmarking, particularly because that's kind of the stage the project's currently at.
Right.
So that
No,
would
I
be,
agree.
and it's something that we have really good expertise in as a team. So I think that would make a lot of sense. And but we just need to sufficiently change the title and the kind of the summary to make sure that it's very clear that there is not as much overlap. I'm not sure it's enough money. I'd forgotten that it's 100K over 2 years, or one, like it's not, it's really small, it's a really small pot. So I'm not sure that's enough to do chemistry for degraders, to be honest.
Well,
I'm
I
not sure
mean,
it's just feasible.
but certainly on the modeling side and to support a postdoc and maybe some money for the AI component, I think we can certainly carve that out.
Okay. Does anyone have any other comments or suggestions on that?
From Variational's point of view, if you want us to participate in this, just let us know what you need from us. That's all.
Yeah,
Yeah,
I think
so
that could be really helpful.
yeah. Yeah, so we'll Rachel and I can put that together and flip that to Pete just for any comments that you might have.
Sounds great.
Cool. So we had some back and forth on this Horizons grant. I don't know if Carlo is still on the line. I know he had to drop off, but Rachel and I were discussing about it yesterday, and it would be a challenge not only to put an application in, but whether or not the scope of the Horizon grant, whether or not we would even be eligible for that grant. So I think between the two of us, we're thinking it's probably a bit too much work and low probability that one, we're even eligible, and two, that we would have much of a opportunity to tap into those funds. But if Carlo or Maria have any additional thoughts, if they have experience with these Horizon grants, we'd love to hear that.
Hi, can you hear me?
Yes.
Yes.
So I don't have experience with these grants, and I can confirm that we need to do a lot of upfront work, and also it would be important to be supported by administrative offices. So it's a lot of work, which I personally think I should start one day to work on these horizons network. So we have a network, a European network, which also, and an international, more extended network from SBMA. But I'm not sure that SBMA would be, focusing only on SBMA would be enough competitive for getting the grant. So I don't know. I don't know.
I would
I
say
think
that,
the KDA is a good
yeah.
opportunity, because maybe even if not this year, maybe next year, or sooner or later, you may get enough data to get the grant, and the MDA also is a good one. The AFM, the National Teleton Foundation, is another very good agency that gives grants also internationally, not only to France. So that's another opportunity. Now, I don't know the deadline. I think is past for this year, but maybe for the next year. It's a super good one.
Sorry, what's the name of that organization, Miriam? I
It's
didn't
the Association from the French Teleton Association.
Okay,
AFM.
right.
AFM.
Yeah, AFM.
Is the French Teleton. It's like our Fondazione Teleton, or like the MDA in US.
Okay. All right, I'll look into that.
But
Thank
I
you so much.
I can send you, but you can find them.
Yeah.
And they really get every year like a budget of 80-90 millions euros. So they have enough to also sponsor international projects, not only French projects, like is in Italy, that we can't afford international grants.
Okay, that's great to know. Thank you so much. I think so. The good news, bad news on the horizon is I think this particular one, it's not really. I think the SBMA scope is not going to be fundable. But the good news is that I did talk to the folks at SGC Frankfurt. They have a lot of experience submitting horizon grants, and they said if we find something that comes up in the future that where we think we're a better fit, then they would be happy to support the application. They've submitted a ton of these and been successful. So
that's good to know.
So
yeah, that's great. That's
if we find one that looks
important.
good, then we can do it. They have a lot of experience and know how to, because they said it's non-trivial to submit. So I think once we find one that's a better fit, then I think we should go for it. But not for this one, I don't think. I think when I dove into the hundreds and hundreds of pages of like back documents about all the requirements, I was like, I don't think we're going to be funded on this particular call, but I'll just keep an eye out and see and bounce it off them because they have a lot of experience. So
yeah,
great.
That's fantastic.
I'll keep you posted.
Okay, cool. And then the last one that just came out recently is this NSERC Horizons grant, and I think we'd have to kind of look as a group of at least in Canada who would be eligible as a applicant or a co-applicant. We would need, in order to take advantage of the maximum funding, we need at least 3 applicants. And Rachel, I don't think you are eligible because you already hold a grant. I don't. Matthew and Mark. Unfortunately, Mark couldn't make it, and I'm not sure whether or not they have grants or would be eligible for this grant either. So I think without Matthew and Mark on the line, it's kind of difficult to see if we would be. We'd have to maybe look outside the group that we have now, and we don't really have much time for that. Whether or not we could find somebody else to co-apply for this. But I think one thing that certainly this project would resonate with the NSERC Horizon is the last bullet point there, because our research does involve Indigenous populations and communities.
Okay. Yeah, we can check. I can follow up with Mathieu and Mark to see whether they still hold. I know Mathieu did hold a discovery grant. I'm not sure if when it expires or when it's renewing.
Yeah.
So. Okay, we'll try and figure that out.
And these, I said, but this could be potentially up to a 5-year grant, right?
Yeah, I believe so.
Yeah. Okay, so I guess we'll follow up with Mark and Matthew and see if there's a possibility that we could put something in for this NSERC Horizon grant. Okay, so that's pretty much all I have for today. So I think to talk about next steps, obviously we have some chemistry that's going to be started in the next few weeks. So compounds will start to trickle in over the next month or two. I think we could probably hold off meeting again, not monthly, but again in 2 months once we actually have some compounds made. But kind of going back to what I said early on, that I think we should do additional modeling. So I think maybe we can have a separate short meeting with the chemists and Matthew to put together another list of ideas for modeling, not only for potentially work to be done at UHN, but having a list that I can put for Charles River. And when they actually have resources, like she said, we could potentially have resources available in the next 2 months. That if we have compounds for them to make, then they could pretty much get started right away. Okay. Any additional comments from anyone? All right. Well, this is, I mean, it's exciting. We're actually starting to get some work, some traction on some work. So results will start to come in. But yeah, so I think I'll set out a potential, if this time works well for everyone. I guess that would be in sometime in July, towards the end of July on a Wednesday, same time. If that doesn't work for everyone, we'll see what we can do to set up the next meeting.
Sounds good.
Okay.
Thank you.
Great to
Thank
see
you.
all. Yep.
Thank you.
All right.
See you
Bye,
next week,
everyone.
Maria.
Bye.
Nice
Take
to
care.
see you all again, and looking forward for the next meeting.
Great. Thank you. Bye-bye.
Bye.